Hello viewers, welcome to the course of Introductory Plant Pathology. Today we will be discussing about the plant pathogen bacteria, and in bacteria we will be discussing about the diagnosis, vegetative reproduction and taxonomy. First of all, we will discuss about the symptomatology in the bacteria, that is what type of symptoms usually we see in the bacteria affected plants and especially our crop plants. Symptoms of bacterial disease are extremely varied and usually characteristics of the particular pathogen. And here we discuss about the most common types of symptoms we see in the plants. The spots on the leaves and fruit, blights or deadening of tissues on leaves, stems or tree trunks, rots of many parts of the plant, usually root or tubers, and wilts due to plugging of vascular tissues. This is the first symptom I am showing you here, about the black spot lesion on peach leaf showing short holes. You are seeing here, these are the holes. These are the infected leaf spots, and these are the holes caused by the bacteria. Holes actually contain the plant pathogenic infection in the plants, and it is nowadays used for taking as a plant resistance. This is the very common disease of caused by bacteria, Xanthomonas citri, and this is known as citrus canker. Here we are seeing the raised irregular brownish spots on the leaves, and fruits are also affected by the disease, and in the market it is a very common sight when we go for the purchasing of vegetables, and we see the corky brownish raised spots on the citrus fruits. Generally we find with the watery spots on the fruits. So this is all caused by the Xanthomonas citri, and citrus canker is the name of the disease. These are the characteristic symptoms, and it helps us for identification of a particular type of bacteria in the plants. Here we are looking into the potato bacterial wilt. As I told you earlier that the wilt are the very important bacterial diseases, because they are caused by the plugging of the vascular tissues, and after bacterial infections the pathogens plug the tissues which conducts the minerals and water translocation in the living plants, and therefore the water and mineral nutrition is not available to the affected plants, and ultimately after infection they show the drooping of the leaves. As you are seeing here, the leaves are showing the drooping site, and it is the potato plant which showing the wilt infection. Secondly, this is the bacterial wilt stream. When we cut the stem of the affected plant and we dip it in the clear water, after a few time we can see a streaming of the bacteria coming out from the cut ends of the stems, and this is the characteristic identification of the bacterial infection in the plants, especially the wilt infections. Then this is the geranium bacterial blight. You are seeing here the bacterial blight spots, which is causing the most severe disease in the geranium, our ornamental plants. Then this is this is very important slide I am showing you here, because this is a common type of thing we can observe in the bacteria affected plants. You see here the bacterial ooze in the drop in the shape of the water droplets, you see here, and it is coming out from within the water soaked lesions affected with the bacterial pathogens. Then the early water soaking in tomato, you can see water soaked spots on the tomato leaves. This is the early symptom, and this is the later symptom. And in the most type of the bacterial infections we find this type of greasy looking bacterial spots having dark black to brownish spots with a yellow halo around it, and this is a late stage of the bacterial infection. We can say that if this type of symptoms are observed in the affected plant, we can say that this is the bacterial infection which is showing in the late stage. This is the spots on the Xylella fastidiosa in the grapes. Actually, Xylella is a type of causal organism which is very difficult to culture artificially on the different artificial nutritive medium. Otherwise, bacteria are easily culturable on the artificial media, but the fastidious nature of bacteria is causing difficulty in culturing of such type of organisms. This I earlier told you about the crown gall. This is a typical crown gall caused by Agrobacterium tumefaciens, and this is the crown gall caused by Agrobacterium vitis in the grape plants, and these are the crown galls which are very akin to the cells as we found in the cancer in the human beings, and a lot of researches are being conducted in the recent times by on the crown galls to study nature of cancer in our human beings. Then we come to the dissemination of the bacteria. When the plants are affected, how they disseminate the disease in the other healthy plants. So, dissemination of plant pathogenic bacteria commonly occurs by wind blown soil that cause plant wounding, particularly during or after rains. Infested or infected seed or any plant part can be sources of bacterial inoculum. Machinery, clothing, packing material and water can also disseminate pathogens, as can insects and birds. This is, I am showing here the typical life cycle of a bacterium, that how the bacteria completes its life cycle. So, here you see the bacteria in the potato. Actually it is showing the potato disease, and this is the potato affected by the bacterial disease. This is the stem and infection of the tuber infected stolon, and this is the cross section of the infected stolon. This is showing the blackening and darkening of the tissues due to the infection, and the infection spread during the storage. When the potato is stored under the warehouses, they are affected bacteria are disseminated in the other healthy tubers. Then desiccated rotten tubers we are seeing here, they are rotted by the pathogens. Then this is the bacterial pathogen, and the bacteria overwinter in insect pupae, rotten vegetables on host plants, and occasionally in soil also. So, they again insect lay egg over potato seed pieces, and the bacteria are inside those eggs. So, contaminated larvae carry the bacteria into the tuber. These are the bacteria, and again the cork layer forms around the infected tissues. You see here, this is the pupae in which the bacteria is there, and the corky type of structures are seen here is caused by the bacterial infection, and then the bacteria is spread from tuber into the young stem and the roots, and likewise the bacteria in intercellular sacs, and they collapse the cell. This type of life cycle is there in the bacteria, and again this is a different type of cycle which showing the maggots in the pupae in the soil, and emerging adults carry the bacteria to other plants, and here we see the plants are infected through soil borne bacteria, and in the here it is in the carrot and Daucus carota, you see here the bacterial infection, and again the bacteria complete the life cycle in this way. Then bacterial classification. As we know, the classification is very important thing in studying the bacteria, because there are a large number of bacterial organisms, and if we classify them in a particular type of order scientifically, then we can easily come to know that what type of bacteria is causing what type of disease in the plants. So, this is the bacterial classification, and the motile cells are having long whip-like flagella which may arise from one or both ends of the cell, they are known as polar, or from all over the cell, they are known as peritrichous. And by the nature of this bacterial phyla, we can identify those type of bacteria. So, based on flagellar arrangement, bacteria are classified into these 6 groups. First is the monotrichous. It is having single polar flagellum at one end. This is found mostly in the xanthomonas. Then amphitrichous, single polar flagellum at both the ends. This is found in the pseudomonas. And then cephalotrichous, several flagella at one end, for example in this pseudomonas fluorescens. Yes, based on flagellar arrangement, in continuation of this type of arrangements, there is lophotrichous bacteria, there are several polar flagella at both the ends. This is found in spirillum. Then subpolar, single subpolar flagellum, this is found in agrobacterium. Then peritrichous, the flagella are all over the cell, for example in Erwinia, and many bacteria have filamentous appendages called fimbriae or pili. And this picture is showing the arrangement of flagella on bacteria in general. This is the monotrichous having the polar type of flagella, lophotrichous, so many flagella are there, peritrichous on all the cell surfaces, and the amphitrichous on both the ends of the bacterial cell. Then I am trying to showing here the types of flagella looking in under the microscope, because for seeing the bacterial cells we have to have so many arrangements in the microscope. This is the dark field microscopy where we will look into the bacteria and we will find this type of bacterial cell under the microscope. Here it is the phase contrast photomicrography where the bacterial cell will look like this, and this is the peritrichous, polar and lophotrichous type of bacterial cell you are seeing here. Then structurally a bacterial cell can be divided into 5 regions. First is surface appendages, that is flagella and pili. Then surface adherents, capsules and slime layers. The capsule in the outermost layer and composed of dye or polysaccharide that forms a gelatinous slime layer around the cell wall. Then cell wall, it provides shape to the cell and protects underlying protoplasm having cytoplasm, chromatin, vacuoles and globules, etc. On the basis of 2 types of chemical composition of cell wall, bacteria are grouped into 2 as gram positive and gram negative. Most of the plant pathogenic bacteria are either gram positive, which are classified within the phylum Actinobacteria, where cells appear purple in color, or gram negative in the phylum Proteobacteria, where cells appear red in color with specific stains when viewed at 1000x magnification with a light microscope. The different colors largely reflect differences in stain retention by the respective cell walls of the bacteria during the staining process. So, here it is the difference between the gram positive and gram negative. It is the slide showing you the when we flood the heat fixed smear of bacteria with crystal violet for 1 minute, then all cells become purple in color. Then add the iodine solution for 3 minutes, all the cell remain purple. Then step 3 is the decolorize with alcohol briefly with about 20 seconds, and gram positive cells are purple while gram negative cells are colorless. And counter stain with safranin for about 1 to 2 minutes, you see the gram positive, this is the gram positive cells are purple, whereas the gram negative cells are pink to red in color. And this shows the how under the normal compound microscope we see the slide of positive and negative bacteria. Then cytoplasm and organelles. They contain soluble cytoplasmic constituents, nucleoid, mesosomes, ribosomes, lamellae or thylakoid or vesicles, and some reserve materials like granules. Then special structures are there in some bacteria. They form sporulation structures, and most characteristic spore structures are endospores, exospores, conidia, spores, that is echinids, myxospores, cysts and dalocyte. Then we divide and the group based on the Burgess manual of the systematical bacteriology, which was found in 1984. Then first we come to the kingdom Prokaryote. Here there is a division 1, which is having the cyanobacteria. The blue-green algae and myxophyceae comes under this division. Then come the kingdom Prokaryote, in which the division 2 is there, which is having bacteria. Part 1 is composed of phototrophic bacteria. It is having 1 order, 3 families and 18 genera. Then part 2, gliding bacteria. It has 2 orders, 8 families and 21 genera. Part 3 is sheathed bacteria. It is having 17 genera. Part 4, budding and appendages bacteria. It is having 17 genera. Part 5 is spirochetes. It is having 1 order, 1 family and 5 genera. And part 6 is spiral and curved bacteria. It is having 1 family and 2 genera. Then part 7 has gram negative aerobic rods and cocci. It has 5 families and 14 genera. Part 8 is composed of gram negative, that is facultative anaerobic rods, 2 families and 17 genera. Part 9 is constituted by gram negative anaerobic bacteria having 1 family and 3 genera. And part 10 is of gram negative cocci and coccobacilli. It is having 1 family and 2 genera. Then part 11 is composed of gram negative anaerobic cocci having 1 family and 3 genera. Part 12 is of gram negative chemolithotropic bacteria, 2 families and 17 genera. And part 13 is of methane producing bacteria having 1 family and 3 genera. Part 14 is of gram positive cocci, 3 family, 12 genera. Part 15 is endospore forming rods and cocci, it is 1 family and 5 genera. And part 16 is gram positive asparaginous rod shaped bacteria, it is having 1 family and 1 genus. Part 17 is of actinomycetes and related organisms. It has 4 genera but not assigned to any family. 1 family with 2 genera, 1 order with 8 families and 31 genera. Part 18 is Rickettsias, 2 order, 4 families, 18 genera. Part 19 is of Mycoplasmas, is 1 class, 1 order, 2 families and 2 genera. We come to the vegetative reproduction in bacteria. In bacteria, the vegetative reproduction is through fragmentation, through budding and through binary fission. Then we come to the fragmentation. Mostly under unfavorable conditions, bacterial protoplasm undergoes a process of division or compartmentalization. This results in subsequent fragmentation, forming minute bodies called gonidia. Under favorable conditions, each gonidium grows to a new bacterium. It becomes apparent that prior to fragmentation, the bacterial genome has to undergo repeated replication so that each fragment gets a copy of it. Then we come to the budding. As we have seen in the fungi, which is having the yeast like structures, and yeast is also vegetatively reproduced through budding. In case of bacterial cells, budding is also there, and in this case the bacterial cell is showing the small protuberance called bud, and this develop at one end of the cell as we are seeing here, and genome replication follows, and one copy of the genome gets into the bud, and then the bud enlarges, eventually become a daughter cell and get separated from the parent cell, and they become two. Then we come to the binary fission. Under favorable conditions, fission is a common phenomenon. Here the cell divides into two similar daughter cells. The bacterial chromosomes attached to the cell membrane and replicates to the bacterial chromosomes. As the cell enlarges, the daughter chromosomes get separated. A cross wall is formed between the separating daughter chromosomes, and it divides the cell into two daughter cells which grow to maturity within 30 minutes. And here this is how the prokaryotic chromosome is duplicated and then continue to grow in the cell, and this forms the division into the two cells. We discussed about different types of symptoms caused by bacterial disease in plants, and we also tried to find out that how we can diagnose the plant diseases caused by different types of phytopathogenic bacteria in plants. Secondly, we today discussed about the classification in detail, that how we can classify on the basis of flagella and the other arrangements in the bacterial cell. And lastly, we discussed about the vegetative reproduction in the bacterial cell, in which the budding and fission and the lastly the binary fusion is discussed.